Good morning, Pallai madam. So please start your live.
A very good morning to invited guest speakers, respected Principal of G. Pulla Reddy College of Pharmacy. A very good morning to invited guest speakers, respected Principal of G. Pulla Reddy College of Pharmacy, Dr. B. Madhav Reddy Garu, President IPA Telangana State Branch, Dr. B. Prabhashankar Garu, and all the registered eminent faculty members. On behalf of G. Pulla Reddy College of Pharmacy, Management and Faculty, I, Pallavi Kanagala, working as Assistant Professor in the Department of Pharmaceutics, feel privileged to extend my hearty welcome to you all to the e-FDP in research updates in pharmaceutical formulations and analytical method development organized by the Department of Pharmaceutics and Pharmaceutical Analysis of GPRCP in association with IPA Telangana State Branch. In this turbulent time, when the whole world is challenged with the pandemic, we have committed ourselves to the safety of the participants. Hence, this online faculty development program has been designed in a comprehensive way to support the industrial and technical aspects of the faculty without hindering the safety and health of the participants. Technology enhanced innovative learning at our institute is founded on a value system sustained by the learning partnership between the teachers and the students. In this note, let us all seek the blessings of Almighty by lighting the lamp and with a prayer song. I invite Ms. Archana Reddy, Assistant Professor of G. Pulla Reddy College of Pharmacy, to invoke the program with a prayer song. Over to you, Archana.
Good morning, one and all. Myself, K. Archana Reddy. Good morning, one and all. Myself, K. Archana Reddy, working as an assistant professor in G. Pulla Reddy College of Pharmacy.
महागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि महागणपतिम् महादेवसुतम् महादेवसुतम् गुरुगुहानुतम् महादेवसुतम् गुरुगुहानुतम् मारकोटि प्रकाशं शान्तम् महाकाव्यनाटकादिप्रियम् महाकाव्यनाटकादिप्रियम् मूषिकवाहनमोदकप्रियम् महाकाव्यनाटकादिप्रियम् मूषिकवाहनमोदकप्रियम् महागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि महागणपतिम्.
Thank you.
Thank you, Archana. It's my pleasure to invite Dr. B. Madhav Reddy Garu, Principal of G. Pulla Reddy College of Pharmacy, to officiate the program with his welcome note. Sir, welcome, sir.
Yeah, very good morning to all, and welcome to the first day speaker, Mr. Partha Mukherjee, and his colleague also, Mr. Narayan Srivastav. And I also welcome all the participants to this webinar. So, in fact, we have started a series of webinars, and this is the second webinar being conducted on behalf of the two departments, Department of Pharmaceutics and Department of Pharmaceutical Analysis. Earlier, we have conducted one webinar by the Department of Pharmacy Practice during the June 25th to 30 June. And this is the second one. Though we are at different places, but still with the help of these ICT tools, we are able to conduct this webinar, and we are able to communicate the knowledge to the various people, and this is how we are spreading the knowledge and gaining the knowledge. As a part of this webinar, and today we are having a very good speaker from the United States Pharmacopoeia India Limited, Mr. Partha Mukherjee. Though we use so many substances, and it is all the time the assays and other characters or standards or in comparison to some substances, and what are those substances, and they are the standard reference substances. And today it is very fortunate to start this webinar with such a kind of a very important topic, that is the uh, reference standards development and characterization. So these few words, I welcome Mr. Dr. Partha Mukherjee, sir. And before that, I think Dr. Padmavati will give some welcome address. Thank you.
Thank you, sir. I'm pleased to introduce the organizing secretaries of this FDP, Professor Dr. Y. Padmavati, Department of Pharmaceutical Analysis, Professor Dr. K. Lata, Department of Pharmaceutics. And I am glad to introduce the members of this organizing committee, Dr. D. Prashanti, Mr. N. Raghavendra Babu, Mr. P. Ravi Kumar, Mr. Sheikh Nasheed Basha, and myself. So I request Dr. Y. Padmavati ma'am and Dr. Lata ma'am to proceed with the welcome note. And I hand over the session to Dr. Y. Padmavati ma'am. Ma'am, please carry on.
Thank you, Pallavi. A very good afternoon, very good morning to everyone. I welcome you all for the 6 days virtual faculty development program jointly organized by Department of Pharmaceutical Analysis and Department of Pharmaceutics in association with IPA Telangana State Branch. UG Pulla Reddy College of Pharmacy reached our 25 years milestone with good academic excellence. 2019-2020 academic year is our silver jubilee year. Coming to our department, Pharmaceutical Analysis Department was started and established in the year 2012. 6 batches of students successfully completed their PG program and are well placed in respectable organizations across the globe. Our laboratory is well equipped with all the instruments and with good infrastructure. So, coming to our prime research areas of interest, we develop spectrophotometric methods using chemometric reagents, multi-component analysis by spectroscopy, HPLC methods for formulations, biological samples, food products, herbal products. We also do bioanalytical method development and validation for pharmacokinetic studies of in-house formulations developed by Pharmaceutics Department. With this brief speech, I conclude with a quote by Brian Herbert: "The capacity to learn is gift. The ability to learn is skill. The willingness to learn is choice." Thanking you all for choosing our e-FDP program as a platform to learn. I wish you to have a great learning experience for all these 6 days. Thank you. Now I request Dr. K. Lata, Professor, to proceed with her welcome note. Over to you, Lata.
Thank you, Pallavi, for setting the ground. Thank you, ma'am, for giving me the opportunity. Very good morning to one and all. On behalf of G. Pulla Reddy College of Pharmacy, I, Dr. Lata, Professor, Department of Pharmaceutics, and one of the organizing secretaries of the program, take the privilege of inviting you all for the online faculty development program. Late G. Pulla Reddy Garu, a great visionary, set up this prestigious institution, G. Pulla Reddy College of Pharmacy, to impart quality education. Post-graduation program of pharmaceutics was established in the year 2008, and the department is equipped with all the necessary resources. It is indeed pleasure that faculty and students are doing exceptionally well and always at their best, both in academics and co-curricular activities. This resonates as an outcome by university ranks, presentation of papers in national and international conferences, publication of papers in national and international peer-reviewed journals, filing of patents, publishing the chapters and books as authors, awards and rewards at national and international forum. As on date, 10 batches of students have successfully completed their post-graduation and were absorbed through campus placements. With this brief introduction of the Department of Pharmaceutics, once again, I welcome you all. Without no further delay, we'll move on to the next step. Over to you, Dr. Padmavati ma'am.
Yes, ma'am. Padmavat madam is going to join.
Okay.
Yes. Padmavat madam join.
Yeah, I'm sorry, some technical problem. Now
Yes,
moving
madam.
to the
Please
today's
proceed, madam.
session. Yeah. Now moving to today's session, we have today's speaker, Dr. Partha Mukherjee, Senior Manager, RSC India, USP. He will talk about Introduction to USP, Reference Standard Development and Characterization. You all know the importance of reference standards in analytical work. So now I request my colleague Raghavendra Babu to introduce Dr. Partha Mukherjee to the participants. Over to Raghavendra Babu.
Yeah. Good morning, dear teachers. I am N. Raghavendra Babu, Assistant Professor, G. Pulla Reddy College of Pharmacy. And I am so pleased to be with you today to have the chance to introduce our speaker of today's session, none other than young and dynamic personality, Dr. Partha Mukherjee, Senior Manager, USP. Hearty welcome to you, sir. Dr. Partha completed his MSc and PhD in Analytical Chemistry from University of Kalyani, West Bengal. Dr. Mukherjee has vast experience of 10 years in the industry. He worked for TCG over 6 years and 4 years for Jubilant Life Sciences. He was mostly engaged with R&D and drug discovery. Dr. Mukherjee has experience in the fields of NMR structural characterization and preparative HPLC. From past 3 years, he is working as Senior Manager, Reference Standard Evaluation, USP. He is responsible for reference standard development. Finally, he is also fascinating person and we are all in for treat today. Without further ado, I request Dr. Partha to start the session. Thank you, sir. Over to Partha, sir.
Thank you for the nice introduction. And I'm really being privileged to be invited here. And good morning to all. Good morning to respected principal, sir. Good morning to all faculties. And let me start my session with, so today I'm going to present about the introduction of reference standards, where I'm going to say about the reference standard development process in USP, as well as how do we characterize reference standards. So myself is Partha Mukherjee. I'm working as a senior manager in USP India Private Limited, and my responsibility is to develop reference standards in USP. Yeah, I'm welcoming you all to this presentation. So today I am going to speak about, this is the agenda for today's speech. So today I'm going to speak about the reference standards or USP reference standards, where I'm going to say what are the materials and substances we are using to develop reference standards, what are the definitions, and what is the importance of USP reference standards. And also we'll shortly talk about USP as a whole, its mission and its vision. Next, we'll talk about how does USP develop reference standards. There are multiple steps involved, starting from the monograph development and to the release of the reference standards to the inventory, but the use of the user and scientists in their laboratories. So we'll discuss each and every steps very in detail. So next we'll see USP General Chapter 11, which says about USP reference standards. And it tells about the definitions, handling and storage conditions, and how to effectively use USP reference standards. We'll see what are the critical and important points mentioned in the General Chapter 11. Next, we will see what are the precautions to be taken and what are the crucial and critical points to be are important when you are using reference standards in your laboratory. And we'll see next about the uses and importance of USP reference standards from a regulatory point of view. And at the end, I'm happy to take question and answers from the audience. So before moving on, let us see the disclaimer slides, which is very important before starting any presentation from USP or on behalf of USP. Because USP text and publications may have legal implications to US and elsewhere, so their language must stand on its own, and USP does not provide any ex post facto interpretation to any of the party, so leaving the other party in a disadvantageous position. So USP wants that all requirements shall be uniformly and equally distributed to all parties. In addition to that, USP shall not provide any official opinion whether a particular article does or does not comply with the compendial requirements, except as a part of verification program and other conformity assessment programs which are not related to USP standard setting activities. During the course of presentations, I may name few of the equipments which are technically or analytical equipments or instruments, few of the brand names, but those are only for the explanation purpose for any experiments or any procedures. It is not that we are endorsing anything, any brand, and we are saying that any particular brand is only appropriate for the USP procedures and techniques, and this is only for the information purpose. So this course material is USP's property, so duplication without written permission from USP is prohibited. And so we did all our effort to make the presentation perfectly correct, but if you see any mistake, if you identify anything, please write to us in the email ID given below. So let us move into the next slide. I think there is some problem. The slide is not moving.
Sir, go to slideshow, sir. Please once again go to slideshow and move, sir. Yes, sir. Again you go to slideshow, sir. Go click on next, sir.
But none of my windows are working actually.
Yes, sir. Next, sir.
So let me close it and reopen it. Just give me a minute.
Yes, sir. Definitely.
Yeah.
Carry on, sir. So please share your screen, sir, so that I will include in here screen.
Yeah, I'm trying that, but it is not opening. I don't know why.
Yes, sir. Just you minutes, sir, so that we can.
Yeah. Can you see now?
Yes, sir. Yes, sir. I'm adding. Yes, sir.
Yeah, it
Please
is
click
okay,
on
no?
slideshow, sir. Thank you. Thank you, sir. Please proceed, sir.
Yeah, thank you, thank you. Sorry for some technical glitch. Uh, so now let us move on to our first topic, which says what is USP reference standards. So let me go through what are the material and substances we are using to develop USP reference standards. So USP reference standards are highly characterized specimens of drug substances, excipients, in a combination. Drug substances, excipients, it may be impurities. Impurities may be of two types. It is either process impurities or degradation products. Maybe biological materials, food ingredients, dietary supplements, compendium reagents, and also performance test tablets. You all know about the USP prednisone tablet, which is used for the dissolution apparatus verifications. So all the reference standards are rigorously tested in USP laboratories. We also take help sometime from the industry and government scientists, and also testing has been done in some of the government laboratories and contract labs. So it is very important to mention here that the USP reference standards are only intended for use in compendial methods only. And if users and scientists are going to use those reference standards in other methods and techniques, that the level value and all other things are the responsibility rest with the users only. But I would like to say only these reference standards, USP reference standards, are only intended to be used for monograph method mentioned in the USP monographs. So before proceed further, let me say a few words about USP. You all know USP is a 200 years old organization, and its mission is to set public standards to improve the global health. And its vision, let us see about its vision. USP envisions a world where all have access to the high quality and safe medicines. Because we know quality issues affect everyone. Means in many parts of the world, we know about the falsified medicines, and uh, fake medicines are killing. So USP's mission to ensure the safety and efficacy of the medicines is well known all over the world. And by preparing standards or by setting standards, USP ensures the safety and efficacy of the medicines. So USP has two types of standards. One is documentary standards in the form of general notices, general chapters, and monographs. And also USP has physical reference standards, which we are talking about today. So these physical reference standards are required in the test mentioned in the USP monographs. And also, it is worthwhile to mention that USP's diversified leadership is responsible for advancing USP's mission and vision. USP has Council of Experts. In every 5 years, USP's Council of Experts were elected by the convention membership, and they are being selected from various fields of the pharmaceutical industries, and they eventually elected the expert committee members. You know, USP has expert committees in various field of pharmaceuticals, biologics, food ingredients, excipients, and general chapters area. So in chemical medicines, we have majorly 6 expert committees. The responsibility of these expert committees are to oversee the USP standard setting process, and each reference standards and each documentary standards are approved by the USP authority, which is USP Board of Trustees and Council of Experts. So now let us move on to our next slides. So let us see the USP reference standards overview. Currently, USP has more than 3,600 reference standards for use in the full range of compendial tests and procedures, which are mentioned in USP monograph. So let us see a little bit history about USP reference standards. In the year 1926, USP in its publication first mentioned about the future availability of reference standards. And after 10 years, first list of USP reference standards were published with 6 number of standards. From 1936, USP started providing standards to the industries for the safety and efficacy, to ensuring the safety and efficacy of their products. So by the year 2020, we have more than 3,600. The actual number lies around 3,800 reference standards in all range of monograph testing. And there are many more reference standards are in development. So now let us move on to our next slides, where a connection between USP documentary standards and USP reference standards is being explained here. All the USP reference standards are mentioned in USP documentary standards. If you read any monograph of USP, you'll see there is a section called USP Chapter 11, where all the reference standards required for that monograph testings are mentioned. So I can say USP reference standard is an integral part of USP monograph. So this monograph development and USP reference standard development process goes hand in hand. And whenever a monograph becomes official, all the reference standard mentioned under section 11 should be available, or I should say must be available at the time when the monograph becomes official. Otherwise, without any reference standard, monographs will not be official. So it is very important that a monograph and reference standards process goes hand in hand, and at the same time they becomes official. So now let us see what are the types of reference standards USP has. So USP has mainly 2 types of reference standards for compendial uses. One is standards with quantitative applications. You will see the assay standards mentioned in the monograph are required for quantitative application or quantitative analytical technique for doing the assay. And also we have impurity standards, which has quantitative applications in the test of either limit or quantitative impurity testing. And USP also has standards with qualitative applications like NMR standards, IR standards, UV standards, melting point standards. They are being used for comparing your product qualitatively with the USP reference standards. And one of the major applications of qualitative USP reference standards in the system suitability of the chromatography, where sometimes the resolutions or retention times are being compared with the product with the USP reference standards. So those are the types of USP reference standards we offered to the industries. There are also other type of reference standards like melting point reference standards, for example, vanillin, which is being used to evaluate your system is correct or not. There is also prednisone tablet from USP, which is being used for the verification of the dissolution apparatus as per chapter 7.1. So these are the various types of USP reference standards. Now let us move on to our next part of the presentation, which says how does USP develop reference standard? So there are multiple steps involved, as I said earlier. Now let us see what are the steps involved for development of USP reference standards. So you will see in these slides, all the steps are being mentioned here. The reference standards development process starts with the monograph development. It goes to the testing in different USP or outside laboratories. It goes to review of data, and finally, it is released to the inventory for the use of industry and other academics institutions. Now let us move on each and every steps in detail so that you can understand what are the scientific rigor and what are the techniques we are using for characterizing and developing USP reference standards. So the first step of developing USP reference standard is to requirement. Why does a reference standard require in a monograph? So a reference standard is required when a new monograph is being published by USP or being developed by a scientific liaison. I would like to say here, there is a different team which is responsible for developing documentary standards. They are scientific liaisons of USP. They develop monograph, and if in the new monograph a set of reference standards is mentioned, so at that time we start developing our physical reference standards. And as I said, both the process of monograph development and reference standard development goes hand in hand. They go parallel way. Excuse me. So the first point is the new monograph development. The next point is old monograph is there, scientific liaison, and from on the request of industry, a new methodology is introduced in the old monograph, which is also called modernization of monograph. So in the modernization of monograph, there may be a case where a new method is being introduced. For an example, suppose previously there was a TLC test for the impurity. Now stability indicating HPLC method is being introduced in the modernized monograph. And in this HPLC method, there may be a set of reference standards is being mentioned. So in that case, those set of reference standards, yes.
Sir, sorry for interruption. Sir, your slides are not moving, sir. Only reference standards overview, that image is there. Your slides are not moving, sir. Can you please check?
Can you see that? Any new slide? Is it moved from that slide?
No, sir. No, sir. Only reference standard overview is there.
Should I share it again? Stop sharing.
Yes, sir.
What is
Please,
happening? Yeah. Let me do. Now, is it visible to you?
Sir, reference standard overview slide is visible to me now.
Now,
yes,
history?
Okay. Yes, sir. Now it's moving. Thank you, sir. Sorry for interruption. Please
no
proceed.
problem. And so, as I said, these are the different steps and overview of all the steps involved in reference standard development. It starts with monograph development, goes to the testing, and ultimately it is released to the inventory. And as I said, the first step is the new monograph development or a modernization of monograph. So USP has different type of lots. If you go to the USP inventory and iStore, you'll see different lot numbers. So USP has a new lot or fresh lot, which is the first lot of any reference standards. USP may have a continuation lot or replacement lot. A continuation lot or replacement lot is normally provided to the normally released to the inventory when the current lot is being depleted or is being finished or being sold out. So at that time, we develop a continuation or replacement lot. And USP puts all its efforts so that when the current lot is depleted and new lot is introduced, the time is same. There is no back order items in the USP's catalog. That is how. I think I could have explained how why the reference standard development process is initiated. Now let me move on to my next slide. It is the bulk procurement. So when we decided to develop a reference standards, next we will see how we procure the bulk from the industry. So there is a specific team involved here which oversee the bulk procurement process. So we normally looking for the donations from the industry and mainly for the suppliers who are FDA approved. If we don't have any FDA approved supplier, then we'll go for other suppliers. So before procuring that bulk, we have prepared a document which is called bulk procurement specifications. The specifications mentioned in the monograph, we follow the same specification for the procurement of bulk. And we have other processes. As I said, we mainly looking for the donation. Otherwise, if we don't have donation, then we go for procurement or purchase from any of the supplier. Or another process we have a standalone synthetic laboratory in India, USP India site, which is only one synthetic laboratory we have in USP, so which is in India. So they also synthesize bulks for us. They mainly synthesize impurity standards, impurity and limit standards for us. So these are the process involved in the procurement of bulk for the USP reference standards. So after procurement of bulk, now now work is for the team of reference standard scientists. The reference standard scientist is responsible for developing reference standards, and he is responsible for the whole life cycles of the reference standard. So starting from the procurement of the material, our reference standard scientist, he prepares bulk procurement specification. After that, when the bulk arrived at USP site, he starts writing a protocol for testing. So planning for testing is very important, to characterize the reference standards. So when bulk is arrived to the USP site, reference standard scientist starts planning about the testing. So we normally use our USP internal laboratories for testing those proposed bulk of the reference standards. We also take help from contract laboratories in the various parts of the world. And also we use government agencies like very frequently FDA, a laboratory we are used for testing our proposed bulk of reference standards. Also, we use some other industrial laboratories, but it is very critical to mention here all the laboratories we are taking help, they must be ISO 17025 accredited. Otherwise, those data are not acceptable. So now let us move on to my next slide. So after planning for the test protocol or testing, what are the goals of testing? So each bulk of USP reference standards are being tested in multiple laboratories. So normally for the quantitative standards, so we send the samples for the 3 laboratory testing. And what is the goal for this collaborative laboratory study is to confirm the identity and also assess the purity of the material and also determine its suitability of use in the USP monograph. The first, let us see what are the tests we are using for confirming the identity. So there are many tests are mentioned in the monograph, but when we do the characterization of USP reference standards, we do not limit ourselves to the monograph testings only. We test the material with all possible tests and all required tests, and to ensure the proper identity of the material. So for the identity of material, we start with the IR testing, and also to the identification of the functional groups. Then we'll go for the extensive NMR study where the 1D and 2D NMR experiments were done to properly characterize the structure of the material. We also do CHN analysis, specific rotation whenever required. And also, next step, we assess the purity of the material by doing various testing. Like for the organic impurity, we do HPLC method. Not only the method mentioned in the monograph, but also we sometimes use various other methods if required for the proper identification of the impurities of the material. We also do for the moisture analysis and water content, we do current pressure titration, or for the volatiles, we sometimes do LOD, and also we do residual solvent testing by doing the GC headspace analysis. Also, we do the inorganic content of the material by doing the residue on ignitions. So by doing all these things, we assess how much impurity in the various forms present in the material. So finally, we also determined whether this material is suitable for use in the USP monograph or not. So all the relevant testing, like all the system suitability and the methods mentioned in the monograph, all the testing has been done to ensure that the material is suitable for use in its compendial applications. So these are the total testing plan I just would like to explain for characterizing our reference standards. So after this testing plan, we sent all the samples to the various laboratories for testing, and after getting the data from the laboratories, reference standard scientists evaluate all the data and just see whether the laboratories testing, inter-laboratory testing, are close enough to each other, are complying, or at par, are very to each other. They are now reference standard scientist is going to develop a document which ensures the suitability of the material for the compendial users. So actually, the making this data is just like accumulating all the data in a proper format. We have our own internal format to do that. So we are compiling all the data, and after that we send it for the review, so that the reviewers also approve this, so that approve and see all the data evaluation, so that they ensure that this reference standard is suitable for use in its all the compendial applications. So all the compilation of data that also involves for the quantitative standards is level value. If you see the level of USP quantitative standards, you will see there is a level value mentioned for either assay standards or quantitative limit standards. So normally the assigned value is being calculated from the mass balance approach. The reference standard scientist is responsible for that. So he deduct all the impurities from 100%. All the impurities includes organic impurities, residual solvents, moisture, and as well as the inorganic content. So after deducting all these things from the 100%, we got the assigned value of this particular reference standards, which is mentioned in the labels and certificates of the reference standards. So after compiling all the data analysis, all the discussion and justification in a single document, this is very strictly a confidential internal document. This document is sent for review to the various stages of review, and ultimately it is approved by the Council of Experts as well as the expert committee. So as I said, all the first lot of reference standards are approved by the expert committees, and the subsequent lots like replacement lots and continuation lots, our internal review is sufficient to release the reference standards. So we always compare our reference standards when we are releasing to the inventory, all the possible testing and all the possible things, so that we can ensure that these reference standards is suitable for its compendial applications. And also it is worthwhile to mention each reference standards documentation means all the data, everything is going through multiple stages of review by the esteemed reviewers, and sometimes it is also for continuation and replacement loss, sometimes it is also reviewed by the expert committees. However, the first lot of all reference standards is always approved by the expert committees. Now let us move on to our next slide. So this, so after releasing to the inventory, we are not stopping here. Reference standard scientist has also some additional responsibility for the CSU study. CSU is called continued suitability of use. So in USP reference standard, we do not have stability data, stability study, but however, we do the CSU study after interval of 2 to 3 years, depending upon the stability of the material. So we do some of the testing to ensure that that material at that reference standard is still suitable for use in its compendial applications. So in the CSU study, mainly the parameters we check, the identity of the material is confirmed with the IR or UV spectroscopic technique. We also test the material with the stability indicating purity methods, which were tested during the release of the lot. Again, it is tested during the CSU study to make sure that is the impurity level of the reference standards remains constant or within the acceptable range of the assay or limit standards. So we also check volatiles like LOD or KF, and after doing all the tests, we compare this data with the previous data. Previous data means when the data were acquired during the release of the lot, and we decide whether this material is still suitable for use or not. And CSU testing, as I said earlier, is also overseen by the USP reference standard scientist. So now let us move on to our next part of the presentation, which says about the precautions while handling USP reference standards. So this is a typical level of USP reference standards, where you'll see the reference standard name, its catalog number. If it is an impurity standard, then IUPAC name is also mentioned. And there are instruction for use, which is very important, is mentioned in the level. So there are mainly 4 types of uses. One is the as-is basis. So it should be used without any correction or without any treatment. So there is anhydrous basis, where the user needs to correct the weight after doing the KF analysis of the substance. There is a dried basis, where the user is need to correct the weight after doing the LOD from the material, and also dried material. So the user should dry the material before using the preparation of solution or any other compendial uses. So these are the 4 different types of uses in the mentioned in the USP level. This is called the instruction for use. And as you know, as-is basis is the most convenient way to use any reference standards. However, in some of the cases where the volatiles are not constant over the ranges of humidity, as you know, in the different parts of the lab, they have different humidity level. So we have no other choice to give it as an anhydrous basis, dried basis, or dried material, where user needs to perform KF analysis or LOD before doing the quantitative applications or quantitative calculations. So I have actually captured some of the examples, how do we calculate assigned value for the various types of applications of reference standards. The first one is the as-is basis. The first one is the anhydrous basis. As-is basis I already have explained, where all the impurities and volatiles are being deducted from 100% and to get the assigned value. So here in the first example, it is the anhydrous basis, where user needs to perform an KF analysis of the material and correct the weight. So when we establish our assigned value, we do not deduct the KF analysis result from this 100%. So we only deduct the organic impurity, which is found from the chromatographic analysis. We deduct residual solvents and ROI. We keep KF. We do not use KF analysis result in this anhydrous basis assigned value calculation. We give it for the users. When they use the material, they should perform a KF analysis and correct the weight for the quantitative applications. So similarly, there are some other applications, like you may find some of the level of the USP reference standard that there are 2 assigned values mentioned. One is for the chromatographic applications and one is for the spectroscopic or UV spectroscopy application. So for the chromatography applications, we detect all the impurities, organic impurities, water content, and residual solvents so that the material can be used for the chromatographic quantitative applications on as-is basis. And in the second part, for the spectroscopic applications, we do not detect the organic impurity levels because in the spectroscopic application it is not as specific as chromatographic HPLC application. So the impurities is also going to absorb the light. So here during the assigned value calculation, we do not deduct the organic impurity level. So we deduct water content and residual solvents as well as the inorganic content by RY from the 100%. So this is how we establish assigned value for the different type of applications. So a few examples I have additionally. One is for the dried basis. So where in the level it is mentioned the reference standards to be used for the dried basis. So there the user should perform an LOD with the material and correct the wet with the LOD results. So when we establish assigned value for the standards which is going to be used on dried basis, so we do not deduct the LOD value as you see in the slide. We deduct the impurity from the chromatographic analysis and also ROI. So we believe that during the LOD condition, all the volatiles like all the moisture and all the residual solvents are being expelled by the material during the LOD condition. So another, similarly for the dry material use, if you see in the level, the material to be used after drying. So here also we do not deduct the LOD during the assigned value establishment. We only deduct the organic impurity from chromatographic or HPLC, EPLC method and also the inorganic content by RY. So these two examples are very similar, although a little bit different. So I try to explain all the applications of USP and also how do we establish assigned value for the different applications. So now let us move on to our next slide, which says about the critical information which you should see when you start analysis with a USP reference standard. So what are the things you should look into when you read the level of USP reference standard? You should see that USP reference standard name is mentioned. Like here, I have taken an example of USP ritonavir reference standards. So that name should be mentioned in the USP level. Catalog number for each USP reference standard, there is a catalog number. If you go to the USP online catalog, you will see the catalog number for each different reference standards. Also, the package size, how much material inside the vial is there, is also mentioned in the label. And most importantly, the instruction for use, whether it is going to be used on as-is basis, whether it is going to be used on anhydrous basis, or dried basis, or dry material, all the things, every information is mentioned in the label as well as in the certificate. And also, handling and storage condition, which is also important, because if you do not handle and store the USP reference standard as mentioned in the label, it is very much impact your results. The assigned value will not stand if you store the reference standard in a different condition which mentioned in the label. So assigned values will not work there, and you will get the results which is not appropriate. So storage and handling condition is very important, and please strictly follow what is mentioned in the USP level and certificate. Also, the label contains the hazard information. So here, as I said already, the points to be remembered when you read the USP reference standards level, and also observe all the details like name, catalog number, and everything. And it should be and it must be stored as recommended in the level and certificate. Otherwise, any deviation from the storage and handling condition, you will see the atypical observation. Your results will be highly impacted. And when you start using the reference standard before preparing solution, you always go to the USP catalog and you should see whether the particular lot you are going to use, it's still valid for use or not. On each day, on every other day, not every other day, on each day, USP catalog is being updated. So if there is any change in the valid use date by any factor like stability or other things, so USP always updates its reference standards valid use date. So please see the valid use date of a particular lot of reference standards before you're going to start your analysis. So if you have any question regarding the USP reference standard storage, usage, or handling conditions, you may write us to rstech@usc.org. But before writing to rstech, you should go to the usp.org page and go to the frequently asked question where you might have got all of your question to be answered. So these are the precautions to be taken before using USP reference standards. So before using a reference standard, if any compendial test, make sure you're using a valid lot of the material that I have already said. Please, before starting your experiment or analysis, please go to the USP catalog and see that particular lot is still valid for use or not. So if you see, there are two lots mentioned in the USP catalog. One must be the previous lot and one is the current lot. Current lot do not have any valid use date, but the previous lot should have a valid use date. Depending upon the stability of the material, it extends from 6 months to 1 year when the current lot is being released to the inventory. So you also advised to refer to USP General Chapter 11 for all your relevant questions on how to effectively use USP reference standards. I have this overview of USP General Chapter 11 in my presentation. So let us see what are the important points there. They are also advised to go through USP Chapter 659 for additional information on packaging and storage requirements. So that's what I was telling, that I have a short few number of slides where I give you an overview about the USP reference standards, which is Chapter 11 of USP. So USP Chapter 11 overview consists of, it will give you the description of what are the USP reference standards and what they are not, means where you are going to use the USP reference standards and where you should not. And also types of reference standards, as I already said, quantitative, qualitative, performance verification. There are different variety of reference standards are there in USP's catalog. It is also going to give you what are the information on packaging and storage and labeling, what are the critical and crucial information you should read before you start using USP reference standards. And effective use of reference standards, how to use that, how to make solution, how to appropriately do the analysis, everything is mentioned there. So, and also it is, you are advised, all the users and scientists are advised to go to usp.org page and you will find the frequently asked question related to the reference standards and you are advised to go through it. So most of your questions are already answered there. So if you still have any other additional question, you can always write us to rstech@usp.org. So these reference standards, as I already said, are solely given for the use in the compendial testings only. So in other areas, if you want to use, if any scientist or anybody want to use that reference standards in other application other than the USP compendia or monograph, the responsibility is user to how to determine the uses of that reference standard. But USP ensures its reference standards is suitable for its all monograph or compendial applications. And each new reference standards or new use, there is a new term I am using here, which is called new use. The new use is nothing but another program of USP. So this is used for already having a reference standards in our catalog. Suppose a monograph mentioned in the USP's catalog, a monograph is being modernized, and there in the monograph a new test is being introduced where the old reference standards is mentioned. So this old reference standard is being tested with the new method to ensure that this reference standard is suitable for the new use in this new method. So this part of the action of this action of this process in USP is called new use. It is just like using a reference standard already exists in our catalog to be used in our new technology and new method to be proposed in our monograph. And level directions for use are lot specific, means you can see in the multiple lots, the instruction for use and storage condition may change, because as you know, we can procure bulk for different suppliers. Suppose this previous lot was purchased from one supplier, and the next lot is going to be purchased from or procured from another supplier. So sometimes the storage handling condition sometimes changes, and also assigned value is also lot specific. It depends upon the analysis we perform with each lot. So assigned value, storage, and handling condition supposed to be changed on lot to lot basis. As I already discussed, that purity assignments of quantitative USP reference standards are determined, or the assigned value of quantitative USP reference standards are determined by a mass balance approach. And qualitative reference standard doesn't have any assigned value on their level. So this is a legacy or old reference standards, which are very old, which are released before 2004. They do not carry any level value in their level. Although they are quantitative, they do not carry any assigned value in their level. So for users of those reference standards, it is advised to take 100% potency for quantitative application. Excuse me. Basha, my slides is again stopped. I don't know what is happening. Hello, can you hear me? Hello.
Hello, sir. Hello, sir.
Basha, my slides again stopped. I mean, it is not moving.
Yes, sir. Oh, sir, please, same way you proceed, sir.
Stop sharing and share it again.
Yes, sir.
Yes. Mm-hmm.
Yes, sir.
I'm doing that again.
Yes, sir. Sorry for interruption, sir.
No
Maybe
problem.
some technical problem.
Yes.
Yes, sir. It came, sir.
Yeah. Chapter 11.
Yes,
Can you see that? Yeah.
Yes,
So
sir.
let me try to move. Yeah.
Yes, sir.
Now it's moving.
Yes, it's moving. Thank you very much.
Mm-hmm. So all the users and scientists who are using USP reference standards are advised to go usp.org, where you will find all the frequently asked questions related to USP reference standards and many other parameters. So you will find that, and I hope many of your questions will be answered there. So this slide also says that USP's response to COVID-19 situation. We are going through a very unprecedented pandemic situation right now. And USP makes all its effort to make available and ensure all the reference standards should be available in the inventory of the USPs, so that this COVID-19 related medication should not be stopped. All the supply chain should not be interrupted. So this is USP's effort during these COVID-19 situations. All related COVID-19 related medications, all the related reference standards are USP doing all effort to release as soon as possible. So now let us move on to uses and importance of USP reference standards. We'll see some of the regulatory standpoints. So you can see USP reference standards are being used for raw material testing in the pharmaceutical industry. Like API or excipients, they are being qualified against the USP reference standards. As I already said, we have more than 3,600 number of reference standards in variety of field, like in the API standards, limit standards, excipients. And it is also, USP reference standards are also used during the processing of the material. And also, it is very important for the finished product before releasing the batch of any pharmaceutical industry. It should be compared, or it should be quantified against the USP reference standards. And also, during the stability study with the stability indicating method, USP reference standard is very useful for determining the or assigning the stability or evaluating the stability of the material. So now let us see some of the very important points related to our general notices. So I advise all of you who are using USP reference standards in their laboratory to please go through the general notices section of the USP, which is very important. In the general notices section 5.80, you'll find the mention of USP reference standards, and it is said that where USP or NF tests and assays call for the use of USP reference standards, only those results obtained by using specified USP reference standards are used. So it is worthwhile to mention here that our standards are being legally enforced by US Food, Drug and Cosmetics Law of 1938, which was passed by US Congress, which ensures, this law actually ensures the safety and efficacy of all the food, drug and cosmetic products. So this is one of the example where you all know about the 483 citation by FDA. These are the some reasons I try to accumulate why those observations are being made. So the first observation you'll see I highlighted some of the part. So that firm which got this citation, the reference standards they did not obtain the reference standard from any official source. So the response which that industry gave doesn't adequately explains that they used and reference standards from an official sources. So it is very important. It is very important to use reference standards for qualification of your product. Use a reference standards from an official source like USP. Now let us move on to example 2, where it is mentioned it is here that firm is failed to complete the records of testing and standardization of laboratory reference standards. Since many of the pharmaceutical industry laboratory, they use their secondary or working reference standards, but when you are going to use those standards, it must be ensured that those working or secondary standard must be qualified against the official source of primary reference standard. As all the USP reference standards are primary reference standards, so it is very convenient to qualify secondary or working standards in the laboratory to qualify against the primary reference standards provided by the USP. So failing on that, those kind of 480 citation may be observed. So let us move on to next example. Here it is said that the primary reference standards from an accredited institution was not procured. So testing USP reference standard or primary reference standard is very important. As this example, you can see that that particular laboratory did not use a reference standards procured from an official organization like USP. So it is very important to use the primary reference standard procured from official sources. And as I say that all the USP standards are legally enforced. So it is very important for use of, this is one of the very important criteria of using USP reference standards in the laboratory testing and qualifying batch of any pharmaceutical industry. And it is also surveyed, and you will find in our website it is mentioned that it is very easier for any industry it is also surveyed among the industry, and it is verified that it is very easier if you use USP reference standard to qualify your batch with the regulators, and the ANDA failures are very minimal and nominal if you use USP reference standard for qualification of your product. Because all the USP reference standards are primary reference standard, and you have already seen how much scientific and technical rigor we are putting for developing each batch of USP reference standards. So, as you know, all the pharmaceutical industries which are releasing their product to the market for consumption for the public will are actually trying to putting best quality products to improve the health of patients. And also USP reference standards. If you see USP reference standards, the foundation of quality which USP is trying to make is USP reference standards is one of the very strong bond for this quality chain, which is trying to make along with the other industries to improve the public health. But together with the industry, people, and academics and USP, we can build a strong quality change with the help of the standards USP provides, with the help of all the experts in the various fields of pharmacy, and USP's mission and vision to see a world where each patient and each people has the same access to high quality medicine is going to be fulfilled only if we all go together and build a strong quality chain. So I have that's all this much actually for my presentation. So if I'd be very happy to take questions on my presentations. And before I stop, I should thank to all of you, especially to Basha. During this presentation, I faced so many technical glitches. So thanks to you, Basha, for helping me out. And thanks for inviting me. I am very much honored to present my presentation to the institution like Pulla Reddy, Dr. Pulla Reddy. So thank you all.
Yeah, thank you, Dr. Padda. Thank you very much. So it's a nice session. So really we have all the information. Now I request Ravi Kumar to put thanks note to the speaker. Before that, we'll have one question. We'll go through that. K. Sridevi, so she asked like, she's asking to brief out the challenges in preparing reference standards for natural products. So you discussed impurities, biologics and all. She has a question like, so can we do development of reference standards for natural products?
USP has in its portfolio, USP has reference standards for the biologics. So, can you name any natural product particularly you want to do, or can you go through the USP biologics reference standard catalog whether this reference standard is already exists or not? If you are not satisfied with the answer, you can always write to us for which reference standards you are looking for.
That's all, sir. From my side, is it an expensive procedure, that reference standard development and characterization, right? When you synthesize a new drug substance.
So are you talking about cost involved there?
Yeah, cost and the time.
Yes, cost and time are very significant here. So for cost wise, I cannot say you exact figure, it is confidential, but I can say huge cost is involved because we procure bulk from an industry, we send them for the collaborative study for different laboratories, and all the laboratory, they charge for their own charges for the testing those standards, and then again we pack those reference standards in our inventory and then release. So the total process actually requires huge cost. And if I say the time, it is almost taking 2 years, almost 24 months to to establish or develop each batch of reference standards. Okay. You can see how much lengthy the process is.
Yeah. One more question we have, sir. Does the USP provide certificate of analysis for its reference standards?
Actually, we provide certificate, but not certificate of analysis specifically. So, but we provide our certificate where the level information, structure, case number, chemical information is mentioned, but not certificate of analysis. Do not disclose the testing results to our customer because of the scientific and legal confidentiality. But we do provide a certificate where the level claim and all the relevant informations are mentioned.
Okay, sir. Thank you. Now I request Ravi Kumar to put thanks note to the speaker, Dr. Partha.
Good morning, sir. Good morning, one and all. Dr. Partha Mukherjee, sir, you have given a very great start to 5 days e-FD program of G. Pulla Reddy College of Pharmacy. We are very much thankful to you, and I want to express my heartfelt thanks on behalf of G. Pulla Reddy College of Pharmacy as well as the organizing committee of this 5 days e-FD program. Sir, myself, I am very much enlightened with this knowledge because the teachers are having many doubts regarding the monograph development and the reference standard development. Generally, we teach to the students monograph development only, but how they are developed hand in hand along simultaneously, you clearly explained and you have provided crystal clear information regarding the steps involved in the development and characterization, and especially the precautions to be followed in observing the reference standard label, dried basis, undried basis, as it is basis. This is very much useful to all the teachers. So we will convey the same to our students in our classes, sir, because this is most important for the students who are going to enter into quality control as well as analytical development. For freshers, if they are having this information, they will get through the interviews very easily. So no doubt, sir, we are enlightened with the new knowledge today, and your talk has boosted up with new knowledge, as well as we got good encouragement, so that we will move to the next sessions and tomorrow sessions with the same enthusiasm. So thank you once again, sir. Thank you.
Thank you. I'm honored.
Over to Basha.
Sir, before closing the session, sir, we have one more question by the one more participant.
Yeah.
Sir, by the Tahsil Sharma, what is the criteria for other funding agencies like you told that you're looking for donations from industries that are FDA approved?
So as I said, there is we have a specific team of procurement specialists. They looking for the procurements of bulk from various industries. And sometimes we, in a good relation with the industry, some industry came forward to donate bulk for us. And for that donation, they will get some benefit out of it. Means they get some free vials of reference standards, and it is also mentioned that from which and it is also some benefit to the industry. So it is a good relation between USP and many industries where we get donations. For most of our reference standards are started with the donations only. So this is going on with a good relationship.
Yeah.
So many industry actually came forward to donate the bulks for developing reference standards. So so many of the industries see that actually their bulk is being developed as a reference standards. So they are happy to see that.
Yes, sir. Thank you very much, sir, for explaining all the details. We are very thankful to you be here, your valuable time for spending here with our institution. Thank you very much, sir. So now, dear participants, we have a few remarks regarding the registration form, registration links. So please visit G. Pulla Reddy College of Pharmacy, GPRCP Hyderabad YouTube channel for all the details related to the all the 6 days. Link is same. For assessment come feedback form, please visit our college website gprcp.ac.in and subscribe to GPRCP Hyderabad YouTube channel for more updates. And the assessment come feedback form will be active after 1 hour of the session, and it will be active for 24 hours only. So on behalf of organizing committee, we thankful to all the participants, and at the same time all the organizers for conducting e-FDP on e-FDP, which is conducted by Department of Pharmaceutics and Pharmaceutical Analysis, organized by G. Pulla Reddy College of Pharmacy, and in association with Indian Pharmaceutical Association, Telangana State Branch. And thank you very much. So myself, Sheikh Nasib Basha, signing off today, and the same enthusiastic will join tomorrow morning at the same 10:30 a.m. onwards. Thank you very much.